winning the fight. The resistance is greater than the force generated by the muscle. In an isotonic contraction, the force generated by the muscle exceeds the resistance. And as soon as you overcome the resistance, what happens to the force for the remainder of that contraction? Does it continue to rise?
No, it reaches
peak tension.
It's not max tension. What if it's a small weight that you're overcoming? And you could produce more force for a bigger weight, but you don't need to.
Tension remains constant.
There's no name for what you're reaching. The point is, as soon as you overcome the load, the tension stays the same. That's what happens to the force. It stays the same. And that's where the term isotonic, same tension, comes from. It's a waste of energy to generate any additional force if you've already overcome the load. If you want more force, motor unit recruitment to get more force. So isotonic means same tension, but if you look at the graph, it's not the same tension the whole time. Because at some point, you have to go from 0 to overcoming the load. But once you do, then it's same tension. That's where the term isotonic comes from. And during isotonic contractions, what happens to the length of the muscle?
During isotonic?
May shorten, may not. Depends on the type of isotonic contraction, but you can always say it changes. The length of the muscle changes during isotonic contractions. Isometric, same length, does not change. So, if you put a load in my hand that I can handle, that I can overcome, then the muscle length is changing and the direction of change is shorter. It's getting shorter, shorter, shorter. If I start here and you take this out of my hand and put a 100-pound dumbbell in my hand, it's going to go down. I can't generate enough force to hold it here or bring it up. It's going to go down. But I'm still generating force to lower it so I don't drop it. That's an eccentric contraction. It's still isotonic because I'm generating max force. So tension's not changing. It's all I can do. And the muscle length is changing. And those two things have to be true. No change in tension. There is a change in length. So isotonic, eccentric, length is increasing. Isotonic, concentric, length is decreasing. Either way, length is changing. So when you're going through a range of motion, when you're doing an exercise, you're going through both. Right, so with the bicep curl, I bring it up, isotonic, concentric. I lower it, isotonic, eccentric. Bring it up, isotonic, concentric. If I hold it here, isometric. From a single muscle twitch, how many times are you stimulating the muscle to contract? Once. So what happens to calcium levels from this single twitch, this one stimulus? Well, they go up and then immediately fall. It's a very brief increase in calcium levels. And because calcium levels rise and fall so quickly, interaction between calcium and troponin rises and falls quickly as well. And as soon as they stop interacting, tension starts to fall. As long as they are interacting, increasing at first and then reaching a plateau, tension is going up. Right? Because interaction implies cross-bridge cycling is occurring. As long as troponin is in its changed shape and it keeps tropomyosin out of its inhibitory position, cross-bridge cycling will keep going. But as soon as calcium leaves troponin, and you lose that interaction, slowly but surely, one at a time, tropomyosin molecules move back to their inhibitory position. And tension goes down, 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 down. If I was stimulating a slow twitch fiber, how would that be different, the myogram, than if I was stimulating a fast twitch fiber? Would the latent periods be any different? How about time to peak tension, which is contraction phase? Faster, so a shorter time period to reach peak tension. And how about relaxation? That's faster as well, also a shorter time period. So with shorter contraction and shorter relaxation, what happens to the length of the whole twitch? Shorter. Short means fast. It's a faster contraction. So you've got the red slow twitch, the green fast twitch, and the blue really, really fast twitch. And between gastrocnemius and soleus, gastroc's about half the length. Twice as fast.
The extraocular muscles are those ones that twitch over your eyes?
Not over your eyes, that move your eyes.
What about those twitches that you get in your eyes sometimes? How are those related?
Probably still fast twitch muscles, but twitches happen anywhere.
Extra.
Eyes, arms, legs. I usually get them in my thigh when I get them.
No, that was so weird on your eye when your eye just hurt too much.
But those twitches don't have anything to do with these twitches. So relaxation time isn't so significant. It's really time to peak tension. So if you can shorten the muscle in less time, that means the velocity of shortening is greater. And fast twitch fibers have a faster velocity of shortening than slow twitch fibers. Explanation for the Treppe effect. Treppe means staircase effect. Tension goes up, up, up over those first 3 or 4 twitches. What's the cell doing a little bit better each time? Releasing calcium. Not sure why this happens, but it has to do with calcium release. So they call it warming up, the terminal cisternae. Notice the next stimulus is happening at the end of relaxation. If you had the next stimulus occur during relaxation, what would happen? Wave summation. And if you maintained this frequency over an extended period of time, it wouldn't climb indefinitely. It would hit a plateau, and it would just kind of hang there. And if you increase the frequency a little more, and even less relaxation was occurring, you would get the same thing, except it would build faster, and the plateau would be higher. But you're still not at peak tension. If you want to get the plateau as high as possible, then you need fused or complete tetanus, or what's simply referred to as tetanus. Tetanus is tetanus. Anything less is incomplete tetanus. So the plateau gets a little higher with a higher frequency until you reach peak tension. And the reason this is it for tension, because now you're making all the calcium available. And there's no more you can do than make it all available. Maximal amount of calcium is there. You're getting the maximal interaction between calcium and troponin. There's nowhere to go up from there. But prior to tetanus, calcium levels are not maintained. They keep going up and down. Each time you stimulate again, another release of calcium, but you're giving it too much time to go back into storage. At tetanus, instead of the calcium going up and down, it stays up. The frequency is high enough to keep calcium levels high.
to do with motor units recruited?
No. When we're looking at twitches, that is per muscle fiber. So this is how you get the most tension out of a single muscle fiber. By getting it to contract in a tetanic fashion, using tetanus. A frequency for that muscle fiber that's high enough to get the most tension out of it. So when we recruit muscle fibers, whether it's just a few or all of them, each muscle fiber that we recruit is undergoing tetanic contractions. To get anything less than tetanus, you can only do that experimentally. It doesn't happen in the body. Motor unit recruitment in the body, motor unit recruitment experimentally, very different. So if you've got a fascicle or a muscle belly, and you put some electrodes on it, and you start applying a voltage, how do you get more and more muscle fibers to become excited?
Increase load.
Increase the voltage. So a voltage that doesn't produce a contraction is called subthreshold stimulus, or a subthreshold voltage. When the voltage is high enough to get some amount, minimal force, then it's threshold. And then anything over threshold as force continues to climb is suprathreshold. Still climbing, climbing, climbing. When you hit a plateau, and further increases in force don't give you any more, further increases in voltage don't give you any more force. Maximal stimulation. Subthreshold, threshold, suprathreshold, max.
Was there one higher than the
Supramaximal,
but it's not going to contract?
no more force. And why do you get no more force beyond supra, or beyond max? You've already recruited all of your muscle fibers. And it's not really motor units when it comes to Physio-X, using electrodes. It's just the stronger the voltage on the outside, the further it travels inside, and it activates or excites more and more muscle fibers. Completely different than what the nervous system does. So then we've got a slide for what happens in the body. This is how we increase tension. It's not by changing voltage. It's by changing frequency. Frequency of what? So frequency is, in this case, it's not experimental, right? So it's action potentials per second. That's frequency in the nervous system. Action potentials traveling down what?
The motor neuron.
Upper motor neuron. The frequency of action potentials traveling down the upper motor neuron is what ultimately determines recruitment. Whether you just get some small plus some small, medium, and large motor units. It depends on the frequency coming down those upper motor neurons. So here's a single upper motor neuron that synapses on 3 different lower motor neurons. And the differences between them are their thresholds. This smaller one, which has a smaller receptive area and a smaller cell membrane, has a lower threshold. It doesn't take a really big graded potential to generate an action potential on this neuron. The graded potentials occur in the receptive membrane, which is the cell body and the dendrites sticking out of it. The action potential begins at the start of the axon. This is the beginning of the axon. It's called axon hillock. You have to have a graded potential that's big enough to spread to the axon terminal and be big enough when it reaches the axon terminal to bring the membrane to threshold. And threshold is different for these different neurons. So if you are graphing it, we're at rest, minus 70 millivolts. This is a neuron, not a muscle cell. Muscle's minus 85. Neuron's like minus 70. Threshold for the small motor unit could be minus 65. And for the medium motor unit, could be minus 60. And the large motor unit, minus 55. So if you release a little bit of neurotransmitter and open a few ligand-gated channels at the postsynaptic membrane, and what is the postsynaptic membrane in this case? Which cell has the postsynaptic membrane? The lower motor neuron. Right? We're talking about APs coming down the upper motor neuron, where you have a synapse between it and the lower motor neuron, and getting neurotransmitter released, binding to receptors in the membrane, bringing in sodium to generate a graded potential. So if the frequency is low, then you're releasing some neurotransmitter. Little time goes by, acetylcholinesterase eats some of it up, you release a little more, some time goes by. You're not getting a whole lot of ACH in that synaptic cleft. You're not maximizing the number of channels that can open. You're just opening some of them. So some sodium comes in, giving you a depolarization, relatively weak, a small graded potential. But for this neuron, it's enough to reach the beginning of the axon and generate an action potential. So as soon as action potentials start traveling down the lower motor neuron, what happens? Contraction. You're now recruiting those muscle fibers. If you don't reach threshold, then your graded potential wasn't big enough to generate an action potential. So with a certain frequency, action potentials per second coming down that upper motor neuron, you'll get a certain graded potential. Depending on the size, may or may not reach threshold. But for the small motor units, it doesn't take much. Right? So let's say at 10 action potentials per second, we're reaching threshold for that lower motor neuron of the small motor unit. Then if we increase it from 10 to 20 action potentials per second, less time is going by between the next release of acetylcholine. It starts to accumulate in the synaptic cleft. It's just like twitch summation. Same thing. You know how you build on the previous twitch, sooner and sooner, tension rises greater and greater? When you build on the previous release of neurotransmitter, it starts to accumulate in the synaptic cleft. Now you can open more channels, more sodium comes in, bigger graded potential. So now you're not only reaching threshold for the small motor unit, but now it's big enough to reach threshold for the small and the medium-sized motor unit. Just by going from, let's say, 10 action potentials per second to 20. So at 10 we were getting just the small, at 20 we're getting the small plus the medium. So tension's going up. And then if you need to generate even more force, what's the brain going to do? Send action potentials down the upper motor neuron at an even higher frequency. So you went from 10 to 20 to 40 action potentials per second. And at 40, you reach threshold for the small, the medium, and the large. And this is why you can't ever skip small and go straight to medium or large. If you're generating graded potentials large enough to reach threshold on the medium, then you're already exceeding threshold on the small. Small are always getting activated. So if they're working all the time, should they be slow twitch fibers or fast twitch?
Fast.
Slow twitch. Slow twitch or fatigue resistant. You want fatigue-resistant muscles to be used most of the time. And then on occasion, if you need to generate a lot of force, then you can recruit the larger, but more fatigable fast-twitch fibers. They're larger and faster. They're more powerful. You just can't use them for an extended period of time because they do fatigue easily. But if you think about it, when we generate lots of force, typically we're doing it over a very short period of time. It's to lift a heavy box off the ground. It's to jump or run up some stairs or push a heavy weight. But it's short term, and there's always a rest period.
So faster frequency results in a larger muscle?
Yes.
It always has to go small, medium, large.
Yes.
Like that. So
In that sequence, and you can't skip small. So they get recruited no matter what, and then it's additive. So does this make sense now? Increased action potentials per second frequency from the upper motor neuron. Which lower motor neuron has the highest threshold? Small motor unit. Which lower motor neuron is recruited first? Small motor unit. Highest threshold is large.
Yeah.
Oh, that was the question, huh? I can read. High threshold is last, low threshold is first. With asynchronous motor unit recruitment, you're generating submaximal tension, which means at any given moment you don't have to use all of your muscle fibers. And if that's the case, why use the same ones for the whole contraction? Maybe use a small group that generates enough force for a few seconds or minutes, whatever it takes before they fatigue, and then turn them off and get another group of a similar size, but different fibers. Get those guys contracting. And about the time they fatigue, get a different group. And this is what we do. So the muscle tone of our skeletal muscles, those slight contractions that occur all the time, even at rest, it's asynchronous recruitment of those muscle fibers. And that's why the muscle as a whole doesn't fatigue. You can maintain a standing posture for hours and hours and hours, and those postural muscles won't fatigue. Now, what if they were generating maximal force? Could you still have asynchronous motor unit recruitment if you're generating maximal force? No, because as soon as somebody relaxes, you no longer are generating maximal force. They're all being used, all at the same time.
So this applies to slow twitch.
Asynchronous, slow twitch. Why is tension greatest at optimal length? What's so optimal about optimal length? Maximum amount of overlap. Maximal number of cross bridges can form. Or the best overlap. Optimal, best overlap to give you the greatest number of cross bridges. So potentially, when you're at optimal length, just about every myosin head has an actin binding site that it can interact with. And it's not a single length as much as a small range. So anywhere in this range, you're still at an optimal length. You're still getting the greatest number of cross bridges forming and cycling. On the right side of the graph, why is tension going down? There is overlap. It's not zero tension,
There's less overlap.
but less overlap. Now there's some myosin heads sticking out that don't have anything to bind to. So fewer cross bridges, fewer power strokes, less tension. At zero tension, that would imply there's no cross bridges forming. There's no overlap. And on the left side of the curve, same thing, fewer cross bridges are forming for a different reason. You're not pulling the thin away from the thick, you're actually sliding the thin over each other. And they're blocking their own binding sites. Getting some crowding here. And when you get to the point where the Z line is pushed up against the thick filament, the sarcomeres can't shorten. So there's no more muscle shortening at that point. In the body, we're always pretty close to optimal length. Somewhere between 80 and 120%. So we're in this range here. Effective load on peak tension. Well, as the load increases, tension's going to have to increase with it if you're going to overcome the load. Assuming you can overcome a 5 gram, a 10 gram, and a 15 gram load, each time the muscle changes in length, it's going to change a little bit less. As the muscle has to generate more force to overcome a bigger load, a little bit less change in overall muscle length. Think of it as more effort is going into generating enough force to overcome the load. Less force is going into overcoming. Does that make sense? More effort goes into generating enough force to overcome the load. So once you actually overcome it, there's not as much left, so to speak, to keep shortening. So the time, and thus the overall length of shortening, is reduced. If you can generate 18 grams, you can overcome the 15. Can't overcome the 20, so that becomes isometric, and there's no change in length. There is no point at which you're overcoming the load. Here you overcome it, here you overcome it, here you overcome it. But once you overcome it, the heavier the load, the less time of shortening.
Is it safe to say that the load and tension are directly proportional?
Force-velocity relationship. As force goes up to overcome a heavier load, what happens to velocity?
Slows down.
It goes down. So it's an inverse relationship. With the lightest load, you have the highest velocity. So if we want to see how quickly I can go from this position to this position, start, end, I can do it really fast with no weight in my hand. Then you put a 10-pound, 20, 30, 40, 50. It's going to go slower and slower until I can't actually move it anymore. When the load exceeds the force, shortening velocity is 0. It's just isometric at that point. On the top right, they're showing you that the overall time of shortening is decreased as the load increases. So from 80 to 50 to 40 milliseconds, as the load increases, the time of contraction is reduced. what are all the factors that influence force? Force from a muscle belly, let's say, not an individual muscle fiber. How many motor units you recruit, and thus the size of the fibers that you activate. As you recruit more and more motor units, you're recruiting bigger and bigger muscle fibers. The starting length of the muscle, when it's stimulated to contract, makes a difference. Most tension when length is optimal. And then this one, high frequency of stimulation, wave summation, and tetanus. That's really talking about an individual muscle fiber. How do we get the most force out of an individual muscle fiber? We stimulate it with a frequency high enough to give us fused or complete tetanus. Not a little wave summation, a lot, unfused, it's straight to fused. So there's almost, well, there's no relaxation period. We're not cutting into relaxation, we're stimulating before it even begins. So it's just contraction, contraction, contraction all the way up. During exercise, if you get up out of your seat, you've been resting for a while, metabolic rate in your muscles is nice and steady. It's been making ATP based on those metabolic rates. You get up and you start walking or start sprinting, doesn't matter. You're going to go through the same sequence of changes. Because whether it's walking or running, what happens to the metabolic rate in your muscles? It goes up. It's just a matter of how much of an increase. So as soon as metabolic rate goes up, the current level of ATP resynthesis won't be enough. Because as soon as you start moving, ATP turnover increases. Right? Myosin heads are burning through it faster. You've got to resynthesize it faster. There is no storage of ATP to draw from. There's just the ATP at any given moment that is there in the cytosol. And there's enough there to power contraction for a couple seconds before you would run out. Right? But of course, we don't let that happen. As soon as you start breaking down ATP at a faster rate, you have to start building it up at a faster rate. Now, it takes a while for the aerobic pathways to catch up to the now higher metabolic rate. For two reasons. There's a lot of steps involved, and you've got to rev up the activity of all those steps. So the enzymes are doing their thing faster. And oxygen is required. And the amount of oxygen that the mitochondria are receiving right now while you're at rest is going to have to go up. So who's going to get oxygen to the mitochondria at a faster rate as soon as you get up and start walking or sprinting? Cardiovascular system. Well, the cardiovascular system can't increase blood flow instantaneously. It has to rev up its activity as well. Heart rate has to go up. Force of contraction goes up. Cardiac output as a whole goes up. Cardiac output is liters of blood flow per minute. So the amount of blood that the muscle's getting while you're at rest is going to have to increase. It takes time. It takes time for the cardiovascular system to catch up to the new higher metabolic rate and deliver the extra oxygen needed. So you can't depend on aerobic pathways immediately. Maybe after a minute or so, they catch up. So how do you get your extra ATP during that first minute? So you burn through a couple seconds of available ATP and then phosphagen system. And then about the time you run out of phosphagen system, anaerobic glycolysis, its activity has revved up to the point where it can now take over as the main contributor. And then that's going to last you about 30 seconds or so. So your first 15 seconds stored and phosphagen system. And then for your next 30 to 45 seconds, anaerobic glycolysis. And then as you approach a minute, then aerobic glycolysis and fatty acid metabolism, the aerobic pathways. So the sequence is stored, which we really don't have, phosphagen, anaerobic glycolysis, aerobic glycolysis, aerobic fatty acid metabolism. Why do muscles prefer aerobic fatty acid metabolism over aerobic glycolysis? Per gram of substrate, you get more ATP from fatty acids than you do glucose. And there's another reason. Which is limited, your stores of glucose or your stores of fatty acids? Glucose. You've got enough glucose to last you a few hours, a marathon. You've got enough fatty acids to last you a few weeks. So we would always prefer to use fatty acids. You get more out of it, and you have a bottomless pit of it.
So why, when runners are doing marathons, if they like hit the wall, they're using their glycogen stores, why don't they be able to keep going if then adipose is this?
You have to have Krebs cycle intermediates available to actually use fatty acids as an energy source. And I saw a cool phrase that summed it up long ago in a exercise phys book. Fat is burned in the flame of glucose, something like that, where glycolysis has to be running and producing intermediates that Krebs cycle needs, along with acetyl-CoA from fatty acid metabolism. You can't burn fats in the complete absence of glucose. You can get those intermediates from other sources. Gluconeogenesis, amino acids. So when you start running out of glucose, you start breaking down a lot more protein. And when you run out of stored glucose, the liver can actually make glucose from lactic acid and from certain amino acids. So that's why the protein breakdown becomes so significant during marathon running. I think they say up to like 10 or 15% of your calories can end up coming from protein. But you don't store protein, so that's not a good thing, because that's muscle wasting. You're breaking down protein you need. But what can you do, and what do marathoners do so that they don't literally run out of glucose? Power Goo and Power Bar and Gatorade and some form of glucose. They keep taking it in for the whole run. So usually when they hit the wall, it's physiological fatigue, not solely running out of fuel. That's the sequence. Why is that the sequence? Why is phosphagen system first? Fastest. It can turn over ATP at the fastest rate. Meaning as soon as ATP is broken down into ADP, creatine phosphate can phosphorylate the ADP to turn it back into ATP. So what we're looking at with ATP turnover, hydrolysis, has to keep up with resynthesis. The energy that's released from hydrolysis is used to do work. It's not available to recombine PI and ADP. This energy comes from fuels. Amino acids, fatty acids, glucose. Those are the fuels. When you break down glucose, what is it called? Glycolysis. You can break it down partially in the cytosol without pyruvic acid entering the mitochondria, and therefore without oxygen being present. So that's called anaerobic, without oxygen, glycolysis. But you only get 2 ATP. It's very inefficient. You basically take a 6-carbon glucose molecule, break it in half, and form 2 3-carbon pyruvic acid molecules. And you get 2 ATP in the process. And if oxygen's not available, then pyruvic acid can't go into the mitochondria. And you don't want pyruvic acid accumulating, because then you slow down this pathway. So as you make the product, you want to use it in the mitochondria, Krebs cycle, or you want to remove it. And the way we remove it is to convert it to lactic acid, which can leave the cell. Pyruvic acid doesn't leave the cell. So it's great in that it's fast. It's 10 steps. This is 1 step. This is faster, but after 15, 30 seconds, this catches up. This now is your major contributor. But this is the problem, and that leads to fatigue. So you can do a lower intensity exercise for a longer period of time because you avoid this. If the intensity isn't so high and the metabolic rate in the skeletal muscle isn't so high, the cardiovascular system can actually catch up completely and match oxygen delivery with oxygen consumption. But even when the cardiovascular system is working as hard as it can, if you're asking skeletal muscles to generate max force, cardiovascular system can never keep up. You can't get enough oxygen into the mitochondria to have just aerobic pathways support maximal exercise. Not possible. But mild, moderate exercise, yeah. And so you can just keep going for hours and hours and hours. So which fiber types rely on the aerobic pathways for most of their ATP production? Slow twitch. And that's why slow twitch are considered to be fatigue resistant. They don't generate much lactic acid.
When you say slow twitch, is that the same as slow oxidative?
So you can call them slow twitch, you can call them oxidative, you can call them slow oxidative, you can call them type 1, you can call them red. And each of those names tells you something different about that fiber type. It's red because it has a lot more capillaries, which has hemoglobin, which has a red color. It has a lot more myoglobin, also red color, compared to fast twitch. Fast twitch are pale or white fibers because they lack the capillary density. They lack the myoglobin. It's not that they don't have any. They have a blood supply. But it's not nearly as much as what the red fibers have. Oxidative versus glycolytic. Slow twitch or oxidative, fast twitch, glycolytic. Glycolytic tells you they rely on glycolysis, anaerobic glycolysis. Oxidative, oxidation of these substrates is what's happening in the mitochondria. First through Krebs cycle and then eventually through electron transport chain. Complete oxidation of these substrates, amino acids, fatty acids, pyruvic acid. So saying that they're oxidative cells tells you they use aerobic pathways. They're making ATP in the presence of oxygen. That's aerobic. Fast versus slow. Fast twitch means the twitch is shorter. They reach tension, they reach peak tension at a faster rate. And they also relax at a faster rate. And the reason for that, the version of myosin ATPase. You have isoenzymes. Same enzyme, but a different version of it. Just like isotopes. Same element, different version of that element or atom. And it turns out that the myosin ATPase in fast-twitch fibers works faster than the myosin ATPase in slow-twitch fibers. Now that's why fast-twitch contract at a higher velocity. That's not why fast-twitch are stronger. Why are fast-twitch fibers stronger than slow-twitch? Because they're bigger. Not longer, bigger diameter, which means more myofibrils, which means more sarcomeres, more cross bridges, more tension. So who has more mitochondria? The slow oxidative or the fast glycolytic?
Slow.
Oxidative. More myoglobin? More capillaries? Fatigue resistant? Better for low intensity, prolonged exercise? Slow oxidation. Slower version of myosin ATPase? Slow oxidative. Smaller fibers? Associated with smaller motor units? There you go. As far as training goes, if you want to get bigger muscles, and that's not increasing the number of muscle fibers, that's making the muscle fibers you have bigger diameter. Do you want to stress slow twitch fibers or fast twitch fibers? Fast twitch. And how do you stress fast twitch fibers?
More weight.
With resistance, forcing them to overcome heavy loads.
Is that, I know like runners will run with, like, run with weights. Is that stressing the fast twitch fibers? I mean,
A little more.
Will make you faster.
A little more than if you didn't. They're just trying to burn more calories. So while you're doing that jog, what else could you do just to burn more overall calories while you're doing it?
Really fast.
Stress muscles. You know, so like upper body
Like
muscles.
sprinters running with like a
That's
resistant
different.
parachute
So when you have the little dumbbells, they're actually, if they're just holding them, then it's just adding body weight, right? And that stresses muscles too. But if they're actually using them, then they're just trying to burn more overall calories. They're not going to build muscles with 2-pound dumbbells though. You know, that's for endurance. That's going to get the slow twitch fibers of those muscles. It's going to stress them, not the fast twitch. You got to lift heavy weights to stress the fast twitch. But yeah, the weight packs, the parachute,
That's what I was thinking of.
yeah,
So,
that's going to stress the fast twitch.
so you build muscles because the fibers are bigger,
They do.
and then how? By myoblasts?
Yeah, increasing the number of contractile proteins, making more myofibrils, packing more myofibrils into each fiber, making the fibers bigger. So that the first part of the question is, if you want to build muscle size, what type of training do you need to engage in, right? Resistance training. Which fibers are you stressing with resistance training? Fast twitch. How do they respond to resistance training? How do they respond to being stressed? The way you want to think about this is they do what they do well already, even better. So what are fast twitch fibers already good at? Anaerobic glycolysis. How can you get better at anaerobic glycolysis? Store more glucose. Synthesize more glycolytic enzymes. And you can get more ATP out of that substrate. And they do that. They increase their glycogen stores and the number of glycolytic enzymes. The other thing you do well is generate a lot of force. And how can you do that better? Get bigger. Synthesize more myofibrils. How do you stress slow twitch muscles?
Lighter weight, higher reps.
Submaximal contractions, prolonged contractions. Now, if you're kind of looking at two extremes of resistance training. And if someone, was to ask me, and I got this all the time at the gym, I want to get toned. I want to get in better shape. I don't want to get big muscles. And if it was a female, I'd say, well, you're not going to get big muscles anyway unless you inject testosterone. If it was a male, I'd say, okay, then you want to do higher reps, less weight. And you know, now it's more about endurance. You know, you're going to strengthen the muscles somewhat too, but what you're really going to improve is muscular endurance. If you want to get bigger and stronger, then you need heavy weights. And with heavier weights, fewer reps are going to be possible. And so usually you want to keep your reps, you know, under 10. And then if you're going for more of endurance, toning, more like 20. If you can do more than 20 and you just put the weight down because you hit number 20, you're not going to get anything out of it. That's too light of a weight. So when you hit 20, you should be fatigued. If you're not, you need to use more weight. Slow twitch, endurance exercise, jogging, as opposed to sprinting. You're not going to recruit very many large motor units. You're going to use mainly small because the force you're producing is relatively low. You're just asking those muscles to keep doing this over an extended period of time. So you stress the slow twitch. They adapt. They get better at what they're already good at. What are they already good at? Making ATP aerobically and therefore avoiding fatigue. So how do they get better at making ATP aerobically and avoiding fatigue? More capillaries, more myoglobin, more mitochondria. Everything you need to give the cell more oxygen to make ATP aerobically. More oxidative enzymes in those more mitochondria. So they don't really get any bigger. We're not using them to generate much force in the first place. But they get better at endurance. They last longer.
Is jogging pretty synonymous with the muscle toning in terms of the use of small motor units and slow twitch muscles and that kind of thing?
Versus sprinting to more larger motor units to stress the fast twitch. And when you look at the musculature of a marathoner, they're pretty lean. Compare that to a sprinter. Sprinters, they got big muscles.
Mm-hmm.
Huge hams and quads and glutes, big muscles. From chapter 10, it's a little bit of terminology regarding origin, insertion, agonist, antagonist, synergist, fixator. And then a lot about naming muscles. So if the muscle is temporalis or abdominis, how is that muscle being named? In other words, what does the name tell you about it?
The joint?
Location,
The bone?
body region. Doesn't tell you origin or insertion, doesn't tell you action, it just tells you where to find it. Something called abdominis, you would expect to find abdomen. Something called brachialis, brachium, arm. There's so many. Intercostals, between ribs, temporalis, over temporal bone, frontalis, frontal bone, occipitalis, over the occipital bone. And then relative position, not body region, but relative position, comparing two or more muscles. If one is called superior rectus, then you must have another one called inferior rectus. If there was no inferior, there'd be no superior. They're always going to come in pairs. So one's going to be above, and the other one's going to be below. That's the relative position to each other. Superficialis versus profundus. So you have flexor digitorum superficialis, flexor digitorum profundus. The name tells you, there's a lot in those three parts to the name, but the superficialis part tells you these muscles are in the same area, but one is more superficial, closer to the skin, The other one's deeper. And most likely, you're going to have to move the superficialis away to see the profundus. Externus is synonymous with superficialis. Internus is synonymous with profundus. Intrinsic versus extrinsic. Intrinsic muscles are within. Extrinsic muscles are outside of. So the extrinsic eye muscles are outside of the eyeball. They attach to it and move it. Intrinsic eye muscles are inside of the eyeball. They change the diameter of the pupil.